I'd like to thank the organizers for the invitation. It's been a great visit so far. I hope to tell you about some of the research in my laboratory. My lab focuses mostly on these caveolar microdomains, which were defined in the early 1950s as these little caves that appear in almost every cell type. Here I show EM micrographs of smooth muscle cells where you see these classic flask-like invaginations of the plasma membrane. Interestingly, they're highly accumulating of lipids, cholesterol, glycosphingolipids, and within a small 50 to 100 nanometer span of a membrane, they can house almost every signaling molecule you need to have efficient signaling happen. So for about 40 years, the structures were largely ignored. They were thought of as EM artifacts. It wasn't until 1992 when Richard Anderson at UT Texas discovered the protein, knocked it out, the structures disappeared, and they created some amazing pathologies in mouse models. So the protein itself is quite small, but has a lot of functional domains. It has three C-terminal cysteines that allow it to anchor into the plasma membrane, a transmembrane loop that traverses the inner leaflet of most plasma membrane systems. I'll talk a little bit about mitochondria and other cells or other organelles where we find it. Caveolin scaffolding domain, this is what everyone is focused on in terms of interaction of caveolin proteins with a variety of other signaling molecules. The same domain is contiguous with the oligomerization domain, which allows caveolin to bind to a variety of its own molecules in homo and heterooligomers, forming a large complex. And then the protein can also be post-translationally modified. People have shown phosphorylation sites, sumylation sites, and nitrosylation sites as well. Interestingly, the scaffolding domain is highly conserved among the three caveolin isoforms that have been identified. Cav3 is the predominant form I'll speak about today, which is primarily localized in the cardiac myocyte and the ventricular myocyte. So in terms of caveolin regulation of cell fate, it's important to keep in mind this balance between cell death and cell survival. And in traditional, if you look at across all the different cell types that have been described, a decrease in CAV expression is largely tied to cell death, and an increase in caveolin expression is tied to cell survival. In general, this is a very positive effect for terminally differentiated cells like cardiac myocytes and neurons. It's a negative effect for proliferative cells. And so this is this double-edged sword that caveolin sort of binds to as well. It can cause a lot of neoplasias and cancer development, but at the same time, if you can target it to the right cell, there is potential to survive and adapt to stress. We've gone through a series of papers we've published that describe the general characterization of a heart-specific transgenic mouse. It's a model for studying adaptation to stress. I'll describe a couple of the physiological endpoints that we've characterized over the last 5 to 10 years and sort of where this has led us to sort of accumulate. The initial studies we did out of my interest from grad school was ischemia reperfusion injury. If we take the animals, expose them to an intact in vivo ischemia reperfusion model, 2 hours of ischemia, 30 minutes of ischemia, 2 hours of reperfusion, and we look at infarct size, we see that in a control animal that's exposed to the stressor, there's about a 30 to 40% loss of left ventricle. Interestingly, in an overexpressor without any sort of preconditioning or protective stimulus, there's a protection of the myocardium from ischemic injury, and there doesn't seem to be an adaptive potential if you add on additional protective stimuli such as preconditioning. So what this sort of launched in our minds was this idea that this microdomain and upregulation of this microdomain, particularly in the cardiac myocyte, was a way to adapt the heart to rapid and acute ischemic stress. We went on and subjected this animal to accumulating stress. So this is a TAC model, a 4-week TAC model. We have almost 100% survival in the Cav3 overexpressing animals. Interestingly, what we see in control animals after TAC, there's a dramatic hypertrophic phenotype that's observed, which is largely suppressed in the Cav3 overexpressor. And this is confirmed by LV body weight ratios as well as lung weights and other parameters that we've published in various findings. So in taking them from a heart failure model to a more chronic stressor, this is an epidemic that's facing not only the United States, but also countries like China, the Middle East, all over the world. There's an increasing trend over the last 2 decades or so of a large increase in obesity, across populations, which is then tied to metabolic syndrome and diabetes phenotypes. In the U.S., this comes largely from eating foods like this, high-fat hamburger, lots of sugar, lots of fat. So we wanted to see if we subject our animals to the same stressor, what kind of phenotype would we observe? Interestingly, what we noticed in the transgene negative animals when we put them on a type 2 high-fat diet with a low-dose STZ injection to create a little bit of pancreatic beta cell death, we see an enlarged mouse, so there's a nice body weight gain over a 3-month period. We see the same body weight gain in the CAF3 overexpressor. When we look at their glucose tolerance responses, there's no difference in the transgene negative or the CAF3 overexpressor in response to glucose tolerance. If they give them a glucose load, it takes quite a while for that glucose to come back down to normal. In recapitulation of the human type 2 diabetic model, we see an elevation of insulin levels as well in this animal. So not only do they have this body weight gain, this insulin or glucose intolerance, but they also have this basal increase in insulin, suggestive of insulin resistance. What was striking is that when we looked at cardiac function in these animals, as well as morphometry, we noticed that in the transgene negative animals exposed to the diabetic stress, there's an increase in LV mass that then correlated with a decrease in ejection fraction, a decrease in fractional shortening. In the transgenic overexpressing, in the CAF3 overexpressing animals, what we noticed is that there was a blunting of that hypertrophic response, and we saw a blunting of the decrease in ejection fraction and fractional shortening. So it appears that in a mild or longer term stress, we see again an adaptive potential in cells and cardiac myocytes that are overexpressing caveolin protein. So the final stressor that we've exposed these animals to, and this is a paper that's currently in preparation in the laboratory, is the aging phenomenon. So if we can outlive ischemic disease, if we can outlive heart failure, if we can outlive diabetes, we'll eventually die of old age. And so then the question becomes, what happens with old age? And I've thought about this quite a bit. And if you look at organisms over a span, this is a C. elegans, this is a mouse, humans, and then there's quite long-lived organisms like the giant clam. What happens intrinsically as these systems age is they ultimately fail, and we think that one of the major failure points is the membrane and this ultrastructure that keeps the outside separate from the inside. And then I'll show you some data on where we tie in with this. So we had a model to test this idea. Most aging studies that we do in the laboratory are between 18 and 24 months. So with this CAF3 overexpressing animal, the transgene is always on. We noticed that with the young versus aged animal, there's about a 50% decrease in baseline CAF3 protein in transgene negative animals. With the increased expression in the overexpressors, that expression is maintained throughout the lifetime of this animal, and at 2 years we see a still high level of CAF3 expression. Interestingly, in one parameter of ex vivo Langendorf perfused heart, we're looking at developed pressure here in response to an ischemic event. What we see in a young animal is that they're able to recover about 50% of their baseline function. As a function of just aging and sitting in a cage for 2 years, what happens in old transgene negative animals is they lose about 50% of that reserve capacity that they had when they were young animals. What was striking is that in the young and old CAF3 overexpressing animals, there's a higher recovery of function that's maintained throughout lifespan. This is striking in the sense that we have a physiology here that is stress adaptive at multiple levels, and it seems to track with time as well. Interestingly, and cardiologists will find this of dramatic interest, and there's lots of contention in the laboratory that studies neurobiology and the heart. Which organ's more important, your brain or your heart? And so this data would suggest your heart's more important and able to allow you to live much longer. What we see in these heart-specific CAF3 overexpressor animals is a two-phase response to aging. The first death, primary death in an overexpressor, happens at 27 months, which is nearly a 10-month gain in lifespan for these animals. There's a catch-up phase where the overexpressors and the transgene negatives catch in this middle phase, and then overexpressors that tend to escape this live quite long and well. They walk around in their cages like they're quite young and active. So, in summary of this part of the talk, we see that this heart-specific caveolin 3 expression adapts to a lot of variety of stressors. And so the question becomes, how is this possible at the mechanistic and molecular level? Rong talked about mitochondria in the myocardium. What becomes interesting if you look at even further detail within a cardiac myocyte and the heart structure itself is there's two distinct populations of mitochondria that appear. There's these subsarcolemmal mitochondria, that sit right under the cell surface, the plasma membrane surface, and then there's the interfibrillary mitochondria that sit buried within the myocardium. When we isolate these two subpopulations from mouse hearts, we see that in response to various complex substrates for mitochondria, there's much higher energetic response in the interfibrillary mitochondria, which has been tracked in the literature, that these are the ATP-generating mitochondria in the cell, and less so in the subsarcolemmal mitochondria. Interestingly, when we take subsarcolemmal mitochondria and expose them to various cardiac stressors, in this case a cyclic AMP load, we see a dramatic suppression of mitochondrial function in these particular mitochondria that's tied to an increase in free radical generation as well. And so we went on to further look. PKA is a major regulatory protein in heart systems, and it's tightly regulated in a variety of settings. When we isolate subsarcolemmal mitochondria and interfibrillary mitochondria to look at this one particular signaling conduit that's tied to caveolar microdomains, we see that almost all of the PKA signaling components, the catalytic subunit, the regulatory subunits, as well as anchoring subunits, all localized to these subsarcolemmal mitochondria within the myocardium. So what this sets up is this idea in our minds that the interfibrillary are really responsible for the beat-to-beat generation of energy within a system, and that the target of a lot of the therapies and things we're interested in the laboratory, these signaling or these reactive mitochondria, which is these subsarcolemmal mitochondria. So we've developed this idea of this cellular symbiosis, and this goes back to studies that were published decades ago. We think that there's a symbiosis between these microdomains at the plasma membrane and mitochondria that sit right at the cell surface. So this is a paper. George Peabody discovered these structures in 1953, as I showed you on my first slide. This was a paper he published in 1954, and the title of it is quite striking, the small particulate component of the cytoplasm. He went on to win the Nobel Prize in 1970 for this manuscript where he described the ribosome. In one of the plates in this manuscript, what he failed to notice was this subsarcolemmal caveolar structure in very close proximity to mitochondria. And so we've gone on to quantify this in the heart of mice as well as different adult cardiac myocytes. And what we see is that we see this relationship happen almost very, very frequently in the adult cardiac myocyte. You have this caveolar invaginated structure that sits right in proximity to this mitochondria. And so what we think is set up here is a structure that houses a variety of signaling molecules sitting next to these reactive mitochondria that signal. And it sets up a conduit for this signaling entity. And we've quantified this relationship when we see that this happens quite frequently. We went on to see if this was altered in a preconditioning stimulus. What we see after preconditioning are projections that then connect these membrane microdomains to mitochondria. We see an increase after a preconditioning stimulus, an increase in caveolin protein migrating into the mitochondrial substructure. With gold labeling, we can identify caveolin expression in a variety of mitochondrial membrane systems, not only in the outer membrane, but also in the inner mitochondrial membrane as well. So we went on to test if this idea was palpable in our Cav3 overexpressing animals, the idea being that if this animal is adapted to stress, are its mitochondria better benefited? So we isolated and purified mitochondria from transgene negative and Cav3 overexpressing animals. In fact, what we see happens with preconditioning independent of a protective stimulus. In the overexpressing animals, there's a dramatic increase in caveolin protein within the mitochondria that are adapted to calcium pulsing, calcium swelling, and they have better performance in terms of oxygen consumption with a variety of mitochondrial substrates that they're subjected to. I'll close with this idea. So one of the confirmation experiments we wanted to do was if this notion that caveolin serves as a signaling molecule, and it also serves as a way to organize signaling within a system, could we perturb this to show that there's a direct interaction and effect of this microdomain within the mitochondria? So what we went on to do was, based on the immunogold EM data that showed large accumulation of caveolin-3 within the inner mitochondrial membrane, we created a cell-permeable targeting sequence peptide that has a TAT domain, highly charged peptide domain that allows different proteins to traverse various membrane systems. an intermitochondrial membrane targeting system, then subjected to the caveolin scaffolding domain. The idea being here that if the scaffolding domain binds to itself with this oligomerization complex, and it has a variety of signaling molecules that it's architecturing within the mitochondria, if we load the mitochondria with a high volume of and concentration of this mitochondrial scaffolding domain, will act as a competitive scaffold to compete off these protective proteins, whatever they may be. And the idea is that then will lead to cellular apoptosis. So we were able to test this in a Langendorf perfused heart system. The hearts were equilibrated. They were subjected to peptide delivery for 10 minutes, and then subjected to the standard 25-minute ischemia, 45-minute reperfusion period. And what we see in scrambled peptide-treated animals, as we've seen previously, and I've showed you in our in vivo systems, there's a dramatic adaptation in the Cav3 overexpressing animal relative to the wild-type transgene-negative animals, where we see a better recovery of function. When we treat both of these hearts with the targeted peptide that should disrupt mitochondrial Cav3 anchoring and protein organization, we see that in both of these animals there's a dramatic decline in adaptation to ischemia reperfusion injury. What's even more striking is that in the wild-type animal we see a recovery of function similar to what we see in an old animal. What this suggests in our minds is this idea that there's this constant exchange of caveolin within the plasma membrane and the mitochondria that keeps a heart young, and we can perturb this acutely by antagonizing the cav within the mitochondrial membrane. So I'll conclude with the current work in the laboratory, where large focus within the community has been this microdomain. And I think what's emerging now is that caveolin sits in a variety of other membrane systems within a cell. I've shown you data for the mitochondria. We're also looking into ER and other cell membrane systems where cav may be localized and then ultimately interact and conglomerate to create cell-based stress adaptation. So with that, there's quite a lot of people that have been in the lab over the last decade or so that have contributed to this work. I'll take any questions.